Hello, ladies and gentlemen. Good night, good afternoon, good evening, wherever you are. Here I am with another talk. Actually, I was planning on doing this next week, but just I decided to do it a little bit earlier, and it's again on a very interesting topic: host-microbe interactions. Again, this was part of the course called Experimental Microbiology that was lectured by 3 different labs and 3 different professors. So remember, I talked about a yeast to hybrid system and yeast work and genetic work. And on top of that, we did some lab work regarding photosynthetic bacteria, which is again very important because the things that bacteria produce and make are amazing. So it's important to understand photosynthesis and bacteria. And the last section of the course was about plants and their interactions with the microbes and pathogens and etc. So again, this is a very good study. So let me start with that. Just go down again. So this was prepared by me. This was a homework. Once we have finished the lab work and the experiments, then we would write and submit the lab report following the experiments in the class. So with that said. And let me remind that I like it. I like it the way that the course was structured. So even like many universities, colleges may have problem with the current list of employed professors or faculty, but to make a good class, to make a good course, I think you can combine a couple labs into a lecture and make it a strong class, strong course, educative, so and that helps with the professor, so that and it helps with the students. Students get the best out of that course, and then the professors are not overloaded, with the course load. They get done with the class in maybe a couple of weeks, 2-3 weeks, whatever. So it is a good example, I believe. So, and many of the colleges, universities can take advantage of that. Kind of dissect the course into different sections. Make it more available to the student, to the professors, to the faculty, everybody else. And we need to provide this type of education, hands-on, and regarding current problems, such as we are having in agriculture. And today's lab is actually about agricultural problems and solutions. So let me start with that introduction. Plant cells can be infected by fungi, viruses, and bacteria, especially prokaryotic organisms like bacteria, can cause infectious diseases among economically important plants like potato and Chinese cabbage. And these two make a good salad, you know. If you haven't, try. I mean, that's just like I love it. Like just boil some potatoes and then add some, especially red cabbage. I mean, the mix is great. You will love it. Trust me. You know, with some onions and olive oil and some spicing. You will love it. The potato. Who doesn't like a potato salad anyway? In this study, we will investigate virulence factors of such bacteria. These factors include toxins, quorum sensing. So I'm just gonna basically just read, because just this part is very easy to understand, virulence factors. I mean, that is the key word here. Virulence factors. Like I said, many bacteria or many pathogens. I mean, they don't cause disease for no reason. I mean, disease meaning harming another organism. So what is this? Virulence factors of bacteria. So when they start to secrete or synthesize these toxins, and what is quorum sensing? You know, when do they do it? You know, so this kind of bacterial, communication, quorum sensing, is another term for bacterial communication. Type 3 secretion system. And this is another one, type 3 secretion system, which is like an injection, like a needle injection, imagine, getting vaccinated, or this is something like that, except it is like a poison for the plant tissue, type 3 secretion system, and extracellular polysaccharide degradation enzymes, etc. Because you have to break down the cell wall and the plant material to get the nutrients. I mean, that's what the pathogens want. I mean, it's just bacteria. They want to live and survive, and what they do, they just break down the plant material, cell wall, etc. So plant pathogen, Dicaya dadanti, aka Erwinia crescentemi, because I remember professor was talking about at a congress they changed binomial name. So sometimes they rename the organisms, which is okay. Just at this stage we are not interested in the name, but So plant pathogen, Erwinia crescentemi, is a necrotrophic bacteria which benefit from plant tissue until host organism is dead. I mean, it's like that's a real pathogen. I mean, it kills the plant. It's just simple. It lives on or near plants and enters plant tissue via natural opening like stomata or wounds. Stomata meaning like photosynthetic elements, or wounds. This pathogen secretes pectinase to obtain carbon sources from plant tissue via type 2 secretion system following a type 3 secretion system interactions with the host cell, eventually causing maceration of the plant tissue. The idea is to get some nutrient out of the plant, and in this case this bacteria uses the type 2 secretion system to secrete pectinase, and if necessary another secretion system, type 3 secretion system, uh, and eventually causes the maceration of the plant tissue. Erwinia species can infect different host cells with many different peptide enzymes that they secrete. So just very clear. There is nothing to explain there. Because plants differ, from one another, and bacteria sometimes have different, methodologies or different methods to infect them. Uh, in this case, many different peptide enzymes that they secrete. Pectinase is an enzyme that degrades plant cell wall, thus causing release of nutritional cell content. It's the name of the enzyme, the definition of the enzyme. We will analyze regulatory mechanisms and effects of endopeptide lyase enzyme, which is secreted by Erwinia species during growth on plant tissue products, as well as study type 3 secretion system, genetic components, and regulation of virulence. So just we will study pathogenesis in this lab. Plant cells have developed hypersensitive response mechanisms to eliminate pathogenic infections by sensing the degraded pectin products and necrosis of the infected tissue. And on the other hand, if the plant is attacked by a pathogen, they feel they feel this infection, because certain products are made from the infected tissue. Uh, so plants have their own way of dealing with these infections, and it is important to understand these relations. That's why the lab report is called host-microbe interactions. Regulation of virulence genes is carried out via several external and internal cues, thus allowing either secretion of peptide enzymes via type 2 secretion system or construction of type 3 secretion proteins and the release of effector proteins. So type 2 system is just the release of enzymes. The bacteria just releases enzymes, like when we eat a piece of bread, we secrete saliva, and in the saliva there is called amylase enzyme, which helps with the digestion of starch. So it is similar to that. So but type 3 secretion system. Type 3 secretion system is more like punching the plant tissue. It's more like an injection. It's more it's more complicated. And then there are some other proteins involved. In type 2 secretion system, multiple components located in the inner and outer membrane cleave and terminal sequence of exported proteins such as pectinases. So it's just kind of defining what is type 2 secretion system. Bacteria has this membrane system. Remember. These enzymes, the pectinases, get modified. These enzymes get modified, around the membrane, and then exported eventually. And type 3 secretion system, however, multiple components of inner and outer membranes form a syringe unit. It's more like a structure. Type 3 secretion system is more like a structure. It's like a syringe. that injects effective proteins into plant cells. So it is more like drilling into the cell. It's like it's imagine a bacteria is doing this. I mean, a pathogenic bacteria is like building structures like a syringe and injecting enzymes into plant cells. I mean, so it is interesting. You you don't uh, uh, you don't see this everywhere. Pathogenicity. So you have to respect these pathogens as well, I guess. So materials and methods. Materials and methods, they are provided by instructor, like I mentioned earlier. I mean, our professor provided the plasmids and the media and the cells and the cultures, and then we just followed the procedures and then the instructions, and then tried to finish the experiments. And the first experiment was pectate enzyme activity plate assay. Prepared LB broth culture supernatant of E. coli and Erwinia crescentami were incubated for 20 hours. See, they did that before the lab. I mean, so preparing these courses and is not easy. So they did the pre-work. The assistants do that. The 20 hours, incubated for 20 hours at 28 Celsius in the wells of pectinate activity assay plates. Plates were developed with 5 normal sulfuric acid, Erwinia crescentama culture supernatant, meaning it's just a liquid. Like you grow the culture, you put it into a centrifuge, and you run the centrifuge. You pellet the cells, and then the remaining liquid is called supernatant. I mean, all the stuff in the media, the used media, and whatever, so whatever the cells produced and secreted, and we call that supernatant. So Erwinia crescentami culture supernatant displayed a 2 centimeter heliocentric diffusion pattern around plate wells, figure 1, containing pectate substrate, suggesting pectinase activity of this organism. On the other hand, E. coli culture supernatant displayed no pectinase diffusion pattern around wells, supporting that it doesn't have any pectinase production. So it was a kind of control. They just used 2 strains. One is E. coli, other is Erwinia crescentami culture. And this is, I think this is the date. See, April 13, 2007, Friday, April 13th, 2007. That's the date of the picture. And it said this is the picture, as you see. One has the heliocenter, the enzymatic effect you see on the plate. It degraded the material, the substrate, the enzyme, the pectate enzyme degraded the material in the plate, and you get an image, and this one had nothing. So pectinase production on a plate. This is just so there are not so what is important in this one is just the media. There are not any plant cells here. This is just cells. One is Erwinia and others E. coli growing on a plate containing a substrate, a plant material substrate. And it just shows the enzymatic activity. Uh, you see in one and none in the other. It's called plate assay. And the second assay is called maceration assay. Wild type Erwinia chrysanthemi pectinase deficient mutant strain delta PEL and non-pathogenic E. coli strain was used in a maceration assay. Diameter of maceration area is calculated qualitatively. So this is now maceration assay is completed with live cells. In this case, we use Chinese cabbage, and then we just measured the maceration degree with number of pluses. So see in this one, E. coli didn't cause any maceration, whereas wild type, Erwinia caused this maceration. And then see we did dilution study on this one with the maceration experiment. Just this is 1 to 10 dilution, and this is 1 to 100, 1% dilution, and this is the 1 in 1,000. So this is the most diluted pathogen culture, and you will see differences. So see in the wild type, see regular culture did the maceration, was successful in pathogenicity. It was a successful maceration, and then if you start diluting it, again see the level of maceration decreases from 5 to 4, and if you dilute it 10 more times, it just becomes less macerated. And in the case of 1,000 dilution, using no maceration. So this itself shows you something. I mean, I remember in my previous talks, again, washing is important. So this is actually related to quorum sensing. See if the bacteria is not in large numbers, see this is they are crowded in large numbers. See there is maceration. They are being effective. And as the dilutions increase, the level of maceration decreases. The pathogen becomes less effective. It loses the power to infect. So I think this is clear. So this is and again for this was a mutant, and there was a peculiar situation with this mutant. I will explain that later, but it did the maceration. This is kind of a deleted gene in the pectinase system, and it did have maceration, in the original culture, and then when you start to increase the dilutions, it did still have maceration impact. So compared to the wild type, so this mutant for some reason, was still effective in maceration, meaning maybe they kept communicating. See, the dilutions did not, reduce. See, this one was maybe see quorum sensing. This is what we call quorum sensing, bacterial communication. As they lose their numbers, they just become less effective. But in this mutant, see, it just kept it kept the maceration property. So it's interesting. Maybe so. It's just something to consider and try to understand why just this special mutant. Maybe there is more work needed to understand. Again, comparing wild type. This is, I think, the second wild type was the second wild type was a lab culture. Because even if you get the cell from nature, once you start to grow it in the lab, that becomes the different strain. It still could be wild type, but still so lab culture wild type, so it had the maceration even in low dilution. So it is interesting to see these results, but this is the most striking result. I mean, this is just just regular Erwinia chrysanthemi, and then with the dilution, it loses the maceration power, which is important, and which is true for most of the bacteria. Just true for most of the bacteria. So that's why we have to wash our hands. Washing is important. Cleaning, washing is important. It just applies to surfaces. Anything. Just make sure so you can reduce the number of pathogens, and that helps you. Even if it doesn't completely, just reduce. If it reduces the level of pathogenicity. I mean, compared to the original. See, this is the like full power maceration. So just something to think about. This is just an experiment. I mean, like I said, this is all agricultural experiments. So another enzyme activity assay, pectate enzyme activity spectrophotometric assay. Wild type Erwinia chrysanthemi pectate lyase deficient mutant strain delta pel and non-pathogenic E. coli strain was used in this assay. Bacterial cells were grown in LB liquid broth, that is, and LB PGA media, meaning polyglycerolic acid, to examine extracellular activity. So that's important, extracellular activity. Yeah. So this was this experiment was carried out in a device, and then that and you use spectrophotometry. Culture supernatant. Remember this word. It's the liquid. Well, once you separate the culture from the liquid, that liquid is called supernatant. Is used to measure reaction with 0.05 PGA buffer by using optical density you just you grow the cells, and then you put them in little containers called cuvettes, and then do a reading at 230 nanometer wavelength, and then and compare. It's an incubated mixture. Incubated mixture. Uh, so, so you just measure the culture supernatant with the help of spectrophotometry at 230 nanometers. That's where they do the reading, I guess. Just like I said, this is plant lab. I was in a yeast lab, and this is plant lab. So most of the methodology here. We didn't know, just except we learned in the course and in the lecture. But in our lab, we didn't work with such experiments. So this is the spectrophotometric assay results, pectate enzyme activity. So we'll just go over the results one by one. So just 1, 2, 3, 4, 5, 6 specimens were analyzed, and extracellular activity. See, this is extracellular activity of culture supernatant in different strains of Erwinia crescentemi. So this is another method. See the units. They calculated the slope. They calculated the slope. This and then got the numbers based on the based on the slope. So calculated units. First one is 1.67. Experiment number 2. So first one is just E. coli in liquid broth and PGA, polyglutronic acid. And number two is mutant, delta PEL mutant. It's a mutant in LB and PGA, polyelectronic acid. And then number 3 is a negative slope, as you see, is just E. coli, and in LB, just no PGA. Just number 3 is just a negative slope, just E. coli in liquid broth. And number 4 is Erwinia crescentemi in LB and PGA. This is the chart. Number 5, delta PEL in liquid broth, missing the PGA, so it doesn't have the PGA, just LB liquid broth. And 6, Erwinia crescentemi, 39, 37. This is the lab wild type. This is the lab version, lab wild type, lab strain, still wild type, not mutant, and in LB, in just LB, missing PGA. So when we look at the units, so this is the calculation. 1 unit of PEL activity, pectinase activity, optical density 600. That's the number to calculate the cells. Like you have to have equal amount of cells in every culture, and that's how you calculate optical density. That's called and then most of the time you calculate the live cells with this optical density, and that's the formula. The slope the times 1000 divided by 0.3, and so this is and then you get these values, calculated units, and then the first one, as you see, what is it? E. coli in LG and PGA. which is the control in this case. See that there is a low, unit, 1.67. Not much activity there, not much activity. Number two, see, number two is there's a mutant, delta pel is missing a gene, and the media has LB and PGA, and then you see a higher level, of activity. So it is important, 90.7, much higher than the wild type. So the number 4 is the wild type. So there is something to study about this mutant. So it's not number. So this is the chart actually. You see here, it's a very high level of activity, pectate enzyme activity. So number three is negative, as you see, nothing to calculate. Number four is just 38. For this is the wild type. Number four is Erwinia crescentimi in LB, relatively rich media, NPGA, which material, it's a plant, kind of material. That is. LB is the standard media for many bacteria. Liquid broth. It's a standard media for bacterial cultures. And then when you add PGA, which is a substrate for Erwinia crescentimi, plant pathogen. So this value is 38. Number 4, chart number 4, as you see, you see that there's just from the chart you see there's just a higher level of activity. It's a wild type. Number 5, again low. Number 5, which is LB media, delta pel. See the same mutant, See this was in PGA. See number 2, delta pel in PGA. It gives you 90.7, and then the same thing with just LB, nothing. It just reduced to 5. So something different with this mutant. And number 6, 4.67, again Erwinia crescentimi in LB, no plant material, just LB, Erwinia crescentimi, and not much, So it's interesting, it shows the effect of this plant material in different the strains and in the mutants. So let's continue. We will summarize it at the end. And this is the work with the plasmids and green fluorescent, protein strains, plasmids, and media, detection of transcriptional activities. So this is more like this is just now more detailed work. I work with plasmids, but none of them were expressing GFP. So excuse me, I didn't work with any GFP plasmids. I wish I did. I would love to look at the green fluorescence under the microscope and such. But they had in their lab they had this plasmid probe. So these are Again, just the table shows the strains and plasmids and media. This is LB media, standard bacterial media, and PGA. That's the plant material. So this is just empty plasmid, which has no promoter, and so you don't see any fluorescence. Just this is control, plasmid control. You see no fluorescence at all. So the second part here, we see this promoter measures the activity of the type 3 secretion system, the promoter of the HARP-A gene. Like I said, I don't completely know how these plasmids work, but based on based on what I have learned so far, I'm just trying to interpret and analyze interpret and analyze the results. And then I will talk about the mushrooms based on this. So it's a good model, like I wish I studied with GFP plasmids. So this is gene promoter HARP-A. This is the type 3 secretion gene, and its promoter. So remember, the promoter turns on the gene. The promoter turns on the gene if it is induced. So this is a promoter study. You're trying to see if the promoter is induced or not. And then the plasmids have this particular promoter belonging to the type 3 secretion system. And if the promoter is turned on, if the promoter is induced in the media, it synthesizes this GFP protein, green fluorescent protein, which gives you the color, green color, and then you measure it. So this is mean values. In minimal media, see there's a high expression in regular bacteria. This is mean value. We were able to detect some reading. This is the value 13.95. This is the regular value, like a standard value for bacterial cells, average value for normal cells, and this is the reading we get in minimal media. So the promoter was turned on in minimal media. HARP-A. So and there's not much activity here, as you see in rich media. See, there's LB and there's plant material, the PGA. The type 3 secretion system was not active. That is to understand. See, this is the secretion system with the syringe. It involves proteins, and it's a much complex system than secretion of enzymes like pectinase. This is so this shows that if there is enough nutrient in the environment, in the media, the type 3 secretion system is not utilized, is not employed. Not much activity is not employed. Then similar studies are needed to do that in the mushrooms, and because now in an age we have problems with animal farming and agriculture, and soils are getting drier, and the climate is changing. And mushrooms are a good way of producing protein. Very easy, but we need more plasmids to study their promoters, and then what's happening, in different mediums, and in different composts, and in different substrates. Even to study mushroom pathogenicity, or mushroom virulence, we still need to construct these plasmids based on promoter activity. This is called a reporter plasmid, because at the end, you get a color. It's a reporter gene. GFP is a green fluorescent protein. It gives you the green color if it is synthesized, produced based on the promoter activity. So remember, promoter is the switch box of the gene. So every gene has a switch box. So you can use this system to understand, what goes on in the cell. If the promoter is turned on and not. It doesn't matter if the promoter is turned on. It doesn't matter if it generates GFP protein or some other enzyme. I mean, in normal cells, yes, HARPA gene turns on the type 3 secretion system, but this is experimental. So they just made it. The construct, the plasmid is constructed in a way that this promoter can still be turned on in the right conditions, but turns on a different gene. It turns on a reporter gene. So the first thing to do actually in mushroom labs, and that's why it is important. I want to talk about it. And then yesterday I read a couple papers geared, for Pleurotus ostratus, the oyster mushroom. Getting these plasmids in is not easy because of the chitin, the cell wall. The mushroom mycelium has a thick cell wall, and it is not easy to get the plasmid inside the cell, and then you have to generate something called protoplast, meaning you have to remove the cell wall so that it is easier for the plasmid to get inside. Plasmid is the DNA, and you can construct such a DNA with the promoters and with the special reporter genes on it, but you have to be able to get it inside the cell so that you can do these tests and analyze and what the mushroom is growing on media A, media B, etc. So to be able to do all these tests, you need to construct these type of promoter probe plasmids. That's what they call it. So this is the second plasmid. See, this is again this is a different promoter. See, it's the promoter belonging to the pectinase gene, and the activity of that promoter in liquid broth only is around 5. Significantly, rich media. This is liquid broth. I mean, it's the standard media for bacterial growth, and you don't see much activity. There's 5 point, so that it didn't get induced much in the media containing just liquid broth, no plant material. But in the case of PGA, this same promoter was induced, and the NVC activity, green fluorescent protein, was synthesized based on the activity of this promoter, and which acted on plant material, PGA. And then you see activity, and then the higher average here with the other bacteria. This average standard value. And minimal media, you see lower induction. See, this is rich media, this is minimal media, and this is rich media with the plant material. And that tells you the activity of this promoter is based on plant material. So I think that is. And then let's continue with the results. So as you see, so we need to construct these plasmids in mushrooms for oyster mushroom and king oysters and etc. They have just found the methodology to insert the plasmids. I mean, and later on maybe they will have all these different plasmids, and they will start studying, the promoters. I mean, you have to start somewhere. So then this is the experiment carried out with Chinese cabbage. Let me just see how many minutes we have so far. So 52 minutes passed, so I will try to be fast and summarize. Actually, we are getting towards the end. So this is control plasmid. It doesn't have any promoter, so it's just no protein, just nothing. So Chinese cabbage, which is low in nutrients, Chinese cabbage, and then we will compare it to the potato. See, PT means potato, CC means Chinese cabbage. See, this is just the plasmid without promoter, and no green fluorescent protein. See, type 3 secretion system promoter on Chinese cabbage. We see activity, mean activity is 46, and so it's a relatively higher activity we see on Chinese cabbage. And remember, this is low nutrient. Chinese cabbage, like compared to this, is kind of similar to minimal media, low nutrient Chinese cabbage. It's a plant, it's a leaf, as you see. But it was induced. 13.47. The type 3 secretion system was induced. Remember, this system was like a syringe. So the cell felt that there was no food, no nutrition source. So it decided to synthesize this system, type 3 secretion system, and to get the nutrients out. So we see some activity. And pectinase activity of the Chinese cabbage, we see lower induction. See, pectinase activity of the Chinese cabbage was of the PALD mutant. This was a mutant, remember. And then we see a lower activity. So this is a mutant study. So there was not much activity of the Chinese cabbage with this mutant. Empty plasmid, no promoters on potato. so just a lower value. Uh, and again, this type 3 secretion system promoter, HARP-A promoter on potato, lower induction. Normally, normally standard. This is standard. It should be 70 during high expression. It is 2.73. Why? Because the potato is full of nutrients. Why why do you need to synthesize bunch of proteins to inject and stuff and not necessary. So since the cell employs an economic system, if there is food, why would you try to make extra components and extra systems to get food? There is already there is already source of nutrition. See, in potato, no induction of type 3 secretion system. That is important, and that is true for all populations, not only for pathogens. I mean, if there is food, the virulence is off, as you see. When there is potato, virulence is off. See the virulence. This is the virulence gene, type 3 secretion system, and the most stronger one. When there is potato, when there is food, no virulence. And same thing is true for animals, and for if the lion is well fed, it won't do nothing to you. If the tiger is well fed, it won't do nothing to you. If humans are well fed, if populations are not starving, they will not become virulent. Very simple. That's why this study is very important. Amazing. Very interesting result. See, compared to 70, that's standard, 2.73 on a potato. You don't need to synthesize the type 3 secretion system. And see, for the PLD, this is for the pectinase activity on potato. It is 20.7 compared to 62.79. Let me just go over it to see if I have missed anything. I think it's all said and done. So let me just mention it. This PLD is just a regular promoter of the pectinase enzyme. I mentioned, I guess it was mutant, but it's not, in fact. So so it's just a plasmid measuring the promoter activity of the PEL1 PELD gene, and then we see induction in potato, because potato has this material, plant material, and it was induced. The promoter was induced, and this is type 2 secretion system. And if you compare it to the Chinese cabbage, see, in the Chinese cabbage it was not induced. This same promoter, the pectinase promoter, was not induced in the Chinese cabbage, but it was induced in potato. So that's why these two systems are kind of operating against each other, as you see. See, if one is turned on, the other is turned off. See, in this one, the type 3 secretion system is turned on, and this one, in this one, the pectinase, the extracellular activity. So this the enzymatic activity. Uh, Type 2 secretion system. This is the promoter belonging to the type 2 secretion system. It is turned on. So it's just it works against each other. If one is turned on, the other is turned off. It just so interesting, very interesting. I mentioned it. This was a mutant. It's not. It's just a regular. Uh, just like remember this. I did this experiment like 14 years ago, and then some of the term normally when we see delta, usually means mutation, and just excuse me for that, but just basically what it is, very similar. See, in Chinese cabbage, one is turned on, one is off. And in potato, the type 2 secretion system is turned on, the other one is turned off. It's just the two ways to acquire nutrients. Citrains analyzed with fluorescent activated cell sorter, that's the device for the transcriptional activity after growth in different plant and media. So I'm just gonna basically I have explained pretty much everything, but I'm just gonna summarize now. Discussion. Pectate enzyme activity plate assay data showed that our strain LB media grown Erwinia chrysanthemi secreted pectinase, suggesting nutrient-rich media like LB. Induces pectinase secretion via type 2 secretion system. So in regular media, the regular, I should say, rich media, regular media, LB induces type 2 secretion system, which gives you pectinase. In E. coli, that was a control. Did not produce any pectinase in the same conditions. The E. coli is the control strain here, negative control. Maceration assay displayed interesting results. Delta PEL mutant. That's why I was confused. When you see PEL with the delta, it means a mutant. But so when I was explaining the GFP data, I got confused. So never mind. So the delta PEL mutant showed maceration in the maceration assay. So delta PEL mutant showed maceration of Chinese cabbage. However, it lacks PEL A, PEL E, PEL D, and Y and A, partial deletion of PEM-A. So it lacks a bunch of these genes, this mutant. That's why it was kind of peculiar. The results were kind of weird. It lacks this mutant. This mutant lacks these genes which are required for pectinase secretion. Results suggest an error or background differences on mutant strains. That's why these mutants sometimes they just behave different. So maybe there is more work to be done regarding that mutant. Type 3 secretion and maceration was successful in our wild-type strain and lab-isolated culture. However, dilutions of wild-type cell significantly, see the dilutions of the wild-type cell significantly decreased maceration diameter, and 1000 dilution did not have maceration. So when you dilute the pathogenic culture 1,000 times, it didn't cause any maceration, as an evidence for quorum sensing, meaning the bacteria talk to each other, and if they're not in high numbers, You know, just, they don't cause maceration, meaning you have to wash your vegetables. I mean, it's just plain simple. You know, the moral of the story: wash your vegetables. Okay, you know, I mean, very just because the bacteria or pathogens have this thing called quorum sensing. They talk to each other, and then there's more bacteria, you know, and then it'll be easier for them to, you know, infect the plant tissue and cause rot. I mean, you are paying for grape and, you know, other things, uh, and then you don't want to throw that in the garbage. Uh, So in regulation of the type 3 secretion system, so you see quorum sensing in regulation of the type 3 secretion system in Erwinia chrysanthemi, where concentration of cells is a factor in pathogenesis. So that was my note. So it didn't cause any maceration actually. That's the maceration except. Maybe that was my mistake here. I shouldn't have typed TTS in Erwinia chrysanthemi. It just measures the maceration value, but this particular experiment doesn't measure, you know, in any way. It's just the maceration. I don't know if the maceration is directly related to the you know, type 3 secretion system. Like I said, this is not my field, but if it is related, then maybe it is. It's a relation between quorum sensing and type 3 secretion system. Even the term maceration is like I googled it, you know, because it's been some time, and maceration meaning like the plant tissue gets liquefied, the plant tissue gets softens. I mean, it could be achieved via enzymes or via the type 3 secretion system. Uh, uh, So, but I guess there is a relation between maceration and type 3 secretion system. Like I said, this is not my field, but so the story is that dilution. I mean, if you dilute the cells, it didn't cause maceration. I mean, the plant was intact. It didn't cause any wound. I mean, it's just maceration meaning wound. You know what happens if the plant get infected, like spinach? It becomes mushy, you know. Or if your, you know, the cabbage goes bad, what happens? It gets mushy, you know. It's just that you don't want it. Where concentration of cells is a factor in pathogenesis. So that is the keyword. Concentration of cells is a factor in pathogenesis. That is the keyword. Spectrophotometric assay results were consistent, supporting our previous data. E. coli strains that were grown in LB PGA, it's a controlled strain, nutrient-rich and pectinase-inducing media, because it has this PGA, that induces pectinase, showed positive slope and very low reading. It's a control, you know, it's a E. coli, though, you know, slow units, lower units. Same strain had a negative slope, therefore no pectinase production in media containing only LB. This is just for E. coli, you know, like I said, it's just a wild type E. coli, and it can be found in many agricultural products and etc. E. coli, you know. I mean, in the lab we worked with E. coli strains, but it didn't cause any pathogenicity. It was a mutant strain. It was a different strain. But this particular strain, see, with PGA, with some plant material, it had some activity, some activity, low though. Same strain had a negative slope, therefore no pectinase production in media containing only LB. So E. coli in LB media does not produce any pectinase. It's just suggesting a very slight PGA difference. I guess E. coli gets to utilize plant materials and grows on plant. Delta Pel mutant, pectinase mutant, showed 9.67 units of production in pectinase inducing media, suggesting either error or different background than wild type. So this pectinate mutant was different. It showed very higher level of activity, like for a 9.67 units, compared to 38 in the wild type. But the same mutant activity in LB media without PGA was only 5.3 units, which was lower. Why? It's just like I said, it's a weird mutant. But still shows you the effect of PGA. This is a plant material, plant cell wall material, or some degradation material. PGA. So this mutant wasn't very effective in media with just LB liquid broth, just standard bacterial media without PGA. Erwinia crescentimi strain, this is a lab wild type strain that was cultured in LB media, had a reading of 4.67 units without PGA, lower, supporting previous data that pectinase is not produced significantly in nutrient-rich media lacking PGA. No need to, just synthesize pectinase without PGA. Like I said, cells are smart, organisms. I mean, it's just, if there is no plant material, why would you synthesize pectinase? It's just waste of resources. You have to be economic. All the economists, they have to study biology. The biology is the study of economy. It just, if there's no need, if there's no PGA, no pectinase, why would you? Because you already have the nutrients from the LB media. Transcriptional activities of strains were detected via fluorescence activated cell sorter in different plants and media. This is the GFP work. This method enables quantitative detection of cellular compounds like DNA, protein, or other molecules when these structures express fluorescence signal from each cell that can be captured and analyzed with the help of light sensors. So it's an in vivo experiment, and a lot of the labs should utilize it, especially, like I am saying, the mushroom labs need to start constructing these plasmids with GFP promoters. GFP synthesis. And then if you just if you start to study different promoters, like there are different enzymes that mushrooms utilize to digest wood or sawdust or starch, or just anything. It's so you have quantitative data, so you have better data if you study the promoters. So we need to have those plasmids so that you can measure some activity with the GFP system. So when these structures express fluorescence signal from each cell that can be captured and analyzed with the help of light sensors, At the end, you are measuring the green light, based on the promoter activity. In this experiment, we used RNA mesenchymal cells carrying GFP promoter probe plasmids. See, that's the thing. GFP promoter probe plasmids. We need to make more of these plasmids for oyster mushroom and other mushrooms. Like if you because even for mental health, now that they are giving green light to many mushroom species, so that it could be used instead of antidepressant drugs. Some maybe some mushroom medical, like mushroom industry is growing. Medical mushroom industry is growing, and to understand the medical impact of these genes and or products, maybe we need to construct these plasmids and study more, you know, of those genes, and then so how you can generate more product or medicine, whatever. Uh, for example, I think laustatin in the oyster mushrooms are like it's good for lowering cholesterol. Laustatin just lowers blood cholesterol levels. So if you can like analyze and study the laustatin promoter, with the GFP protein, just an example, or the reishi mushrooms, or other mushrooms, medical mushrooms. We just need these plasmids, GFP promoter probe plasmids. Where transcriptional activities of the DSP, so that's the promoters, TTS effectors, and PELD gene is measured. See, the PELD is the regular gene. in the table, I made a mistake and said that it was a mutant, but the name of the gene is PELD gene, so it's not a delta. So excuse me for that, but I just sometimes stuff happens. So the transcription activities of DSP E and PELD gene is measured. Just reported plasmids are see this one reported plasmid, PELD plasmid, and then the second one HARP A plasmid. So this was the empty plasmid. It doesn't carry any promoters. Cells with control plasmid displayed very low FACS reading. See, no reading at all. Low reading because it doesn't have any promoters to turn on the GFP gene. In all media, including LB, this is a kind of rich media, and rich media plus the plant material, polyglycerolic acid, and minimal media, which is nutrient poor. Cells with probe A, HARP A, this is the indicator promoter for the type 3 secretion system, showed 34.83 total GFP reading of cell in minimal media, and 2.56 in LB, rich media, as well as 3.46 in LB and PGA, again very rich media, suggesting evidence that TTS effectors are not induced in nutrient-rich environments. That's just a take-home message. I mean, the virulence, the most virulent, the system of this bacteria is not induced in nutrient-rich environments. Why would it? Why would it? There is already nutrients in the media. It shouldn't become virulent. GFP mean value for the same strain was also very high, meaning average the GFP fluorescent intensity of GFP expressing bacterial cells. So it just shows you a value for comparison. It was not induced, just very low. Very low reading, 2.56 in LB and then 3.46 in LB plus PGA, compared to 228, means no induction. Data suggests that the PELD transcription, that's for the pectinase, PELD transcription was induced in LB plus PGA media with total to 44 GFP intensity and 251 GFP mean value, providing evidence that pectinase is secreted in nutrient-rich media containing PGA, but not LB only, since total GFP intensity value is 5.8 for such an environment. So the pectinase activity was induced in media with the PGA as expected. That's a plant material, and then which was measured. Providing evidence that pectinase is secreted in nutrient-rich media containing PGA. If there is PGA, then you have the pectinase activity, just simple. But only in LB media is very low. You don't see activation. So PELD was not induced in minimal media. In minimal media lacking PGA. Like I said, if there is no such need for pectinase, if the substrate is not there, why would you synthesize it? It's just simple. But if there is PGA, then you see high intensity, and there is transcription, there's induction. Potato and Chinese cabbage grown strains were measured with facts, and data showed that pectinase was better induced than HARP-A type 3 secretion system in Chinese cabbage. Which is not a nutrient-rich environment, meaning Chinese cabbage. So it is better induced. Was better induced than HARP-A in Chinese cabbage, which is not a nutrient-rich environment. Meaning it was high and similar, both strains suggesting that HARP-A was induced strongly in some of the bacterial cells, since cabbage is not nutrient-rich and can induce CTS, and it was supported by maceration assay. PLD was induced at higher level in potato grown cultures. So just let me read that. So just make sure so make sure it makes sense. So pectinase activity was better induced than HARP-A, that's type 3 secretion in Chinese cabbage. Okay. And mean GFP intensity was high and similar in both strains, suggesting that HARP-A was induced strongly in some of the bacterial cells, since cabbage is not nutrient-rich and can induce TTS. It was supported by maceration. Cabbage, we mentioned earlier, is not nutrient rich, is not nutrient rich, and so it induces the type 3 secretion system. And potato induces the pectinase activity. I think that's what I wanted to say. I guess I'm just this is what I wrote back in the day. So I don't know. Maybe I wrote something wrong or missing. So I'm just trying to make sense of the paragraph. Because I wrote it. I mean I'm just just trying to go back to 14 years and what I was trying to say. Maybe and then I'm just see going back in time and then see maybe some things I didn't write clearly as much. Uh, See, it's reporting is important. I like you just have all these details, and then you don't mention one thing, it just doesn't make sense. But I pretty much explained. So it's different than potato. Potato, the type 2 secretion system is activated. In Chinese cabbage, type 3 secretion system is activated. I mean, just and then we saw that with the promoted activity of HARP and PELD. See, PELD was induced at higher level in potato grown cultures. So this is, like I'm saying, the way I wrote it, good luck with the professors. I mean, it's sometimes it's not easy to read these papers. Imagine like being a professor and trying to read this. I agree. I mean, it just but this is like a state of the art technology. I mean, in those days, I'm trying to understand, and on top of that, we are not a bacterial lab, and we are not doing any work with the plants, and it was just trying to understand. But still, I mean, you get the idea. The pectinase, was induced at higher level in potato grown cultures, 16.2 total GFP intensity, with 60.72 mean average. However, heart rate displayed 3.8 and 3.68 total GFP intensity along with 70, supporting the idea that heart A is relatively induced in small number of cell compared to total. So it was a lower activity for potato because it is nutrient rich, and you don't need to activate the type 3 secretion system for potato. So for potato, so pectinase was induced, and then HARP-A was not. So potato is a nutrient-rich plant compared to cabbage. Therefore, total PELD activity is higher. Yet, type 3 secretion system was still induced in some cells expressing HARP-A. Lower. I mean, you don't see like a complete zero, but the pectinase type 2 system is higher. Pectinase system is higher compared to type 3 secretion system. Experiment was successful, and data supports our hypothesis that the type 3 secretion system is induced in nutrient-poor environments in order to obtain nutrient via pathogenesis and virulence. So this is the end point. After all this mess, I guess that I made the final statement. I guess. So that's the idea, actually. I mean, that is to understand, how the system is induced. So it was successful, and the data supports our hypothesis that the type 3 secretion system is induced in nutrient-poor environments. Virulence, meaning virulence, pathogenicity is induced in nutrient-poor environments in order to obtain nutrient via pathogenesis and virulence. So, ladies and gentlemen, this is very important sentence. I mean, see all this technology, all these plasmids and everything, with all this headache, I'm trying to trust me, the way I wrote this paper, it just God forgive me. But at the end, this is the point, though. The pathogenicity, you know, the, the, the virulence is induced in nutrient poor environments in order to obtain nutrient via pathogenesis and virulence. When you provide food to populations, when you provide the nutrients and food and nutrients, you eliminate pathogenicity. It's just simple. Type 2 secretion system secretes pectinases when there are degraded plant wall products like PGA. The same thing for the mushrooms too. They're not supposedly parasitic. Like for example, the oyster mushroom. You can see them growing on regular live trees, but they can as well grow on dead logs and dead trees and sawdust and other material. It doesn't necessarily need a live tree to survive. So that's what I'm trying to say. Even though the stuff people, organisms, they want to live. They need nutrients, even pathogens. The idea is to get nutrients. So even most of the creams and to stop those infections and etc. Especially for if you look at antifungal creams, look, they are full of nitrogen to stop the virulence. Their nitrogen concentration is slightly higher. You're giving nutrient to the fungi so that it stops the virulence, it stops the pathogenicity. So type 2 secretion system secretes pectinases. Same thing for fungi. Fungi secretes lots of enzymes, lactases and etc. So to degrade the cell wall and everything. Because the cellulase, for example, you need cellulase to degrade the cellulose. Lots of enzymes are necessary. The fungi are amazing. The type 2 secretion system secretes pectinases when there are degraded plant cell wall products like PGA, thus improving energy yield. And it's not virulent in this stage. If there is food, it's not virulent. Type 3 secretion system is not induced where energy and nutrients are available. That's the final point, I guess. And this is the end of the report. So thanks for thanks for listening, and please subscribe to the channel. And I'll try to make more content like this, and like I said, we can learn many things from bacteria, from these cells, the way they operate, and the way the cell economy works. So there are many lessons to be learned, and we need these methods to study the mushrooms, and then so we can increase the yield and make some protein available to hungry people. Thank you very much for listening. Like I said, please subscribe to the channel. Have a great night, day. Thank you very much.